Bueno, ¿qué tal? Dar honor a este tan importante webinar que vamos a presentar, que creemos que merece la longitud porque va a ser, creo, importante escuchar a todas las voces y a todos los expertos, por lo cual, por eso quise ser muy puntual para la presentación mientras todos comienzan a entrar. Bueno, nuevamente nos encontramos. Gracias a todos por venir para poder realmente compartir este momento. Ustedes saben que estamos trabajando muy unidos y fuertemente para tener presente a nuestra querida sociedad en un momento tan especial como es este, por lo cual, como ustedes saben, todo este tiempo hemos hecho muchísimas reuniones, actividades para poder estar presentes. Y para esto también quiero tomar un segundito con la próxima presentación, que es nuestro llamado a la presentación de la próxima secretaria, la CID. Esto es un cargo sumamente importante en el cual tratamos y esperamos que muchos se registren. Hay excelentes candidatos que uno piensa que puede ser. Acá hay una foto de alguien como la doctora Caterine, y también muchas personas a las cuales los invito, porque creo que podrían ser muy buenos secretarios y también en el futuro presidentes de la CID. Así que estamos esperando en esta convocatoria, no se olviden la fecha, así que esperamos que haya muchos candidatos. La próxima, por favor, es que queremos decirles que va a haber nuevamente intérprete, por lo cual ustedes van a poder elegir el idioma en español o en inglés. Por favor, esto es algo que lo hacemos para poder llegar a todos. Sí, por favor. Alfonso, queremos conmemorar el Día Internacional, ¿sí? El 28 de junio, del screening neonatal. El International Neonatal Screening Day es sumamente importante, es pionero en la región Brasil. Tenemos que felicitar al doctor Condino Neto y a todo el equipo que está con él, de inmunólogos brasileros, que están haciendo una labor extraordinaria en el campo de las inmunodeficiencias y sobre todo en el screening neonatal para el diagnóstico precoz de las inmunodeficiencias. Así que nuevamente felicitaciones al grupo de inmunólogos encabezados por el doctor Condino. La próxima, por favor. Bueno, quiero contarles que hemos hecho, ya saben, el curso de citometría para el diagnóstico de las inmunodeficiencias. El diagnóstico, es decir, estuvo dictado por la doctora Isabel Galiar. Ustedes pueden encontrar estos cursos que están en nuestras redes. Por favor, pueden hacerlo y creo que es sumamente importante e interesante, como el diagnóstico genético en síndromes por errores innatos de la inmunidad, dado por la doctora Cecilia Poli, y el simposio que fue extraordinario de ADA2, de DADA, encabezado y coordinado por el doctor Ignacio Uriarte, que fue sumamente provechoso, y también pueden encontrar todas las conferencias grabadas en nuestras páginas. Por favor, la próxima. Bueno, queremos convocar al 15 de julio a que nos acompañen para escuchar realmente qué están haciendo los jóvenes inmunólogos en Latinoamérica. ¿Qué futuro, qué presente, cuál es el campo que pueden desarrollar, cuál es la visión desde el grupo de nuestros jóvenes en la CID, y que debemos hacer un lugar de discusión? ¿Qué le podemos proponer y qué ellos nos proponen? Así que este es nuestro próximo webinar. Los esperamos el 15 de julio del 2021 con nuestro querido Alejandro Palma, que va a ser el coordinador y que creo que va a ser un espacio fantástico. Por favor. Bueno, esperamos que todos nos acompañen en nuestro meeting 2021, que va a ser virtual, encabezado por todo el grupo de inmunólogos chilenos, quienes lo están coordinando, a la cabeza nuestra querida Cecilia Poli, el 13 al 16 de octubre. Seguramente va a ser un encuentro extraordinario, como nos tienen acostumbrados nuestros colegas chilenos, y que, bueno, vamos a tratar de dar lo mejor de nosotros para estar presentes una vez más como sociedad. La próxima, por favor. Saber que tenemos que seguir registrándonos, registrando los pacientes, saben que tenemos que registrar, tenemos una deuda pendiente, ahí la estoy convocando a la doctora Gisela Seminario, que es la presentación de nuestro paper o nuestro trabajo sobre qué pasa en la región con nuestros pacientes, con errores signatos de la inmunidad frente al COVID. Creo que tenemos ya bastante experiencia y que esto lo debemos comunicar y compartir con el resto del mundo. Así que los vuelvo a convocar nuevamente. Gisela, esto es un trabajo que hay que hacer. La próxima, por favor. Bueno, ser parte de nuestra sociedad es algo muy importante, lo demostró la pandemia nos demostró que podemos estar unidos y que podemos hacer trabajos y podemos presentar nuestros resultados, y es algo muy importante, por lo cual le pedimos que se sigan sosteniéndola, inviten a diferentes jóvenes y a diferentes profesionales de cada uno de sus países a ser miembro, y como siempre vamos a tener nuestros encuentros que tienen ese sello característico de calidez y que muy pronto vamos a volver a hacerlo presencial. Y bueno, ahora sí, la idea del día de hoy es presentar una visión de los diferentes países, como en la Argentina, Brasil, y la experiencia puntualmente en los errores innatos que está viendo en Australia por el doctor Teigl y nuestra querida Kate Sullivan en Estados Unidos. Y para esto, Ekaterine Godoris, la profesora del Departamento de Pediatría de la Facultad de Medicina Federal, de la Universidad de Río de Janeiro Federal, y que es una de las referentes en alergia e inmunología de Brasil, va a ser nuestra coordinadora, y le agradezco muchísimo que esté presente y que nos acompañe, como siempre, a nuestra querida Ekaterine. Bueno, todo tuyo, este espacio, Ekaterine. Gracias.
Hey, good evening. Welcome you all. Tonight for this webinar, we'll have speakers that are really very well known and dear to all our clinical immunology community, but a little formality won't do no harm, so I'll introduce them properly. And at the end of the presentations, I will help with the questions. First, we'll have Professor Guilhermo Docena, a CONICET principal researcher, professor of immunology at the National University of La Plata, Argentina. Then we'll have Liliana Berzrodnik, the current president, great president of LASID, also a CONICET researcher and director of the Jeffrey Model Argentina Center. Then Antônio Condino Neto, professor of experimental medicine at the University of São Paulo, a great past president of LASID. Then we'll hear Stuart Tanji, researcher at the Garvan Institute of Medical Research, a professor at the Faculty of Medicine at the University of New South Wales, Sydney, Australia. Then the dear Kate Sullivan, chief of the Allergy and Immunology Division at the Children's Hospital of Philadelphia, past president of CIS. And then Anthony Lacava, Allergy and Immunology Fellow at Penn. He works at the Hospital of the University of Pennsylvania. So welcome you all. And Dr. Professor Docena, please, you have the word.
Buenas noches para muchos, buenas tardes para otros. Realmente para mí es un placer estar aquí compartiendo esta actividad junto con la CID, digamos, una asociación tan importante para todos nosotros. Le quiero agradecer en especial a la doctora Liliana Verosní, quien me ha invitado y para compartirles algunos de los resultados que estamos teniendo en un proyecto que estamos haciendo aquí en Argentina, y también agradecerle a las autoridades de la CID por haberme permitido estar aquí. Entonces, como les decía, yo pertenezco a la Universidad Nacional de La Plata, estoy en un instituto que depende del CONICET y de la universidad, y el año pasado, a comienzo de la pandemia, se creó en la facultad un grupo multidisciplinario que se llama Grupo COVID Exactas, que se ha dedicado a distintos frentes que tienen que ver con la pandemia, cuando nos convocan desde la facultad y desde la universidad para tratar de hacer un aporte desde nuestros lugares de investigación. Nuestro instituto es un instituto que estudia aspectos básicos y aplicados de inmunología, sobre todo de inmunidad de mucosas, y bueno, ahí se forma este grupo y estuvimos todo el año trabajando en el desarrollo de métodos de diagnóstico, de métodos serológicos, y actualmente estamos trabajando en el desarrollo de una vacuna para COVID. A comienzo de año, luego que se inició la etapa de vacunación en nuestro país, nos convoca el Ministerio de Salud de la provincia de Buenos Aires para formar un grupo también multidisciplinario, porque no somos solo inmunólogos, para estudiar la respuesta inmune en individuos vacunados en la provincia de Buenos Aires. Entonces, actualmente la verdad que es un placer enorme estar a 6 meses de haberse iniciado la vacunación y uno poder mostrar esta información, estos números. Pensemos que allá por el mes de noviembre o diciembre, donde se empezaban a autorizar las primeras vacunas, fíjense que hoy tenemos 16 vacunas. Me parece que esta aplicación del New York Times todos la deben conocer, es muy interesante porque esa actualización tiene una actualización prácticamente día a día, y uno realmente se sorprende, por lo menos cuando tengo que dar una charla y me gusta mostrar estos números para resaltar uno de los aspectos muy positivos que nos está dejando esta pandemia, a pesar de todo el desastre sanitario que está haciendo en el mundo, es el desarrollo de nuevas plataformas y mostrar al mundo, a la sociedad en general, cómo la comunidad científica en todo el mundo se ha puesto a disposición para el desarrollo en este caso, de vacunas. Y fíjense que actualmente tenemos prácticamente 120 vacunas en ensayos clínicos, o sea que se están aplicando en individuos. Hay otras 100 que están en etapas preclínicas, como la nuestra aquí en Argentina, junto a otros 5 proyectos. En Argentina hay 6 proyectos de desarrollo de vacunas que están en fase preclínica, y bueno, y actualmente tenemos 16 vacunas que han sido autorizadas, que se están utilizando en todo el mundo. Y fíjense, esta tabla también es real, este gráfico también es realmente impresionante, cómo día a día va aumentando en el mundo la cantidad de dosis que se van aplicando, y actualmente estamos prácticamente el 40% de la población del mundo, 2,7, en realidad es un poquito más hoy, 2,7 billones de dosis de vacunas distribuidas en distintos países. Sin embargo, un problema que se está viendo, que estamos enfrentando, sobre todo los países que estamos en hemisferio sur, es que hay una inequidad muy marcada en la distribución de las vacunas en el planeta, y por eso podemos ver que muchos países tienen porcentajes de su población muy avanzadas en cuanto a las vacunas que han recibido, mientras que, por otro lado, hay otras regiones, como será África y otros países, inclusive en Latinoamérica, sacando Brasil, Argentina y Chile, que el porcentaje de la población vacunada es mucho más reducido. Entonces, rápidamente vemos cómo nos vamos acercando en algunas regiones a lo que uno considera estadísticamente que va a ser la inmunidad de rebaño. Entonces podemos ver casos paradigmáticos como Israel, donde el 61% de la población ha recibido vacunas, prácticamente todas han sido vacunadas con plan completo. También es muy interesante ver en Chile qué es lo que ha ocurrido, lo que está ocurriendo en Latinoamérica, en Inglaterra y en Estados Unidos. Pero bueno, lo impactante y lo interesante que estamos viendo es que ya a partir del 40% de la población vacunada se empiezan a ver cambios en lo que son las curvas de contagio. Entonces, estos ejemplos que yo les mencionaba, fíjense cómo a partir de la vacunación las curvas han cambiado drásticamente, obviamente que dependiendo del país y las condiciones en las cuales pueden acompañar a los planes de vacunación con medidas de protección comunitaria o de cuarentena, eso también tiene un impacto en las curvas. Pero bueno, como les decía al comienzo, allá por noviembre o diciembre, que estábamos en segundas, terceras y hasta cuartas olas de contagios y se empezaba a vacunar en el mundo, no sabíamos cómo iban a responder las distintas poblaciones y qué poder iban a tener de modificar estas curvas de contagio. Entonces, hoy en día no hay ninguna duda que las vacunas son seguras, que las vacunas son eficaces y eficientes y que pueden modificar estas curvas de contagio y probablemente con el correr del tiempo van a modificar el curso de la pandemia. Por lo tanto, la vacunación pasó de ser algo promisorio a ser una gran promesa para poder frenar y controlar esta pandemia. Sin embargo, como les decía, la provisión limitada de vacunas en ciertas regiones y países del planeta han creado que se deban diseñar algunas estrategias alternativas, algunas estrategias sanitarias alternativas de vacunación, y se haya tenido, inclusive actualmente se está, considerando distintas estrategias donde se prioricen grupos que están más expuestos o grupos que tienen mayor riesgo. Nuestro país no escapa a esta situación. Estos son los números en Argentina. Tenemos un 33% de la población que al menos ha recibido una dosis, un muy bajo porcentaje que ha recibido las 2 dosis. Han llegado 18 millones de vacunas. Por suerte, en las últimas semanas esta cinética de llegada de vacunas se ha modificado, pero bueno, todavía nos falta un recorrido como para alcanzar un mayor porcentaje de la población vacunada y tratar de mantener en el tiempo esto que estamos viendo en las últimas 3 semanas en Argentina, que es la contención y la disminución del caso de individuos infectados, de manera que uno pueda llegar a decir que estas vacunas están funcionando en Argentina, a pesar que aquí es muy difícil mantener medidas de restricción. Actualmente en Argentina tenemos 3 vacunas, la Sputnik V, la de Oxford AstraZeneca y la de Sinopharm, pero yo voy a hablar más puntualmente de Sputnik V porque el mayor porcentaje de los individuos ha recibido esta vacuna, sobre todo la primera dosis, y además porque fuera de Rusia es el país que más vacunas de este tipo ha recibido. Entonces, muy rápidamente y a modo de repaso, yo creo que todos saben en qué consiste la vacuna Sputnik V. Es una vacuna con la plataforma a base de ácidos nucleicos, emplea 2 vectores virales que son adenovirus inocuos para el humano porque se les ha reemplazado los genes tempranos de infectividad por los genes que codifican para la proteína S, de manera que cuando este virus, que lleva la información para la producción de este antígeno viral, ingresa a las células de nuestro organismo, mediado por un receptor, este virus puede ingresar prácticamente en todas las células, aquí adentro, bueno, se libera el contenido, el DNA, que codifica para esta proteína y para otras, y se transcribe, se traduce la proteína S, se libera de la célula, también queda anclada, se expresa en el complejo mayor de MHC clase 1, pero lo cierto es que la disponibilidad de esta proteína por fuera de la célula es la que activa el sistema inmune y genera la aparición de anticuerpos. La diferencia entre esta vacuna a vectores virales y otras vacunas, como la de AstraZeneca, por ejemplo, es que emplea 2 vectores distintos, simplemente con la estrategia de, con el primer vector viral se va a generar, por supuesto, la proteína S, los anticuerpos neutralizantes de esta proteína S, pero además no debemos perder de vista que este vector viral, este adenovirus, funciona como un antígeno para las células nuestras y eso genera o induce la aparición de anticuerpos. De manera que cuando en la segunda dosis se da un adenovirus distinto, como el adenovirus 5, estos anticuerpos que se generaron contra el adenovirus 26 no van a reconocer a este vector viral, de manera que la eficiencia de este segundo vector viral para ingresar a las células es mucho mayor que si fuera una segunda dosis de adenovirus 26, por lo cual estaría bloqueado. Estos resultados han sido publicados, los resultados de fase 3 se publicaron en el mes de enero. Ahí lo que se mostró en este trabajo en Lancet es que a través de esta vacuna y este plan de inmunización con 2 dosis separadas por 21 días, genera títulos altos de anticuerpos IgG, específicos de proteína S o de RBD. También genera títulos altos de anticuerpos neutralizantes, si uno compara distintas edades, distintos sexos versus el grupo que recibe placebo. También genera una inmunidad T dependiente con producción de interferón gamma a través de células CD4 y CD8. Y finalmente, si uno ve la curva de acumulación de casos, uno puede ver fácilmente cómo se despega la curva de individuos que reciben placebo versus individuos que reciben vacuna, por lo cual esto significa que la vacuna genera los mecanismos efectores que uno espera y mostró eficacias superiores al 90%. Entonces, a partir que en el mes de fines de diciembre se comienza a vacunar en primera instancia a la población en Argentina, a través del Ministerio de Salud de la provincia de Buenos Aires nos convocan, y por la trayectoria que habíamos tenido durante todo el año colaborando y trabajando con el Ministerio, la Municipalidad y CONICET en el desarrollo de métodos, nos convocan a formar parte de un consorcio junto a otros laboratorios importantes de nuestro país, como la Fundación Instituto Leloir, con la doctora Andrea Gamarnik, y el Instituto INVIRS de la Facultad de Medicina de Buenos Aires, con el doctor Jorge Geffner, de manera de estudiar o monitorear la respuesta inmune en población vacunada en la provincia de Buenos Aires. Entonces, en primera instancia, se reclutaron o se incorporaron individuos que recibieron vacuna Sputnik, 300, un N de 300, luego se incorporaron 100 que recibieron vacuna de Sinopharm, y actualmente se han incluido también individuos que han sido vacunados con Oxford AstraZeneca. Intervienen 7 hospitales públicos de la provincia, y lo que se hace es tomar muestras antes de la vacunación, 21 días después de la primera dosis, 21 días después de la segunda dosis, y luego a los 3, 6 y 12 meses. Es decir, que es un estudio que va a durar un año. Y lo que se determina es la medida de anticuerpos, IgG antiproteína S y RBD. Se realizan titulaciones. También les voy a mostrar que medimos anticuerpos neutralizantes, y a partir del mes 3 estamos empezando a medir inmunidad celular a partir de poblaciones celulares, CD4 y CD8 productores de interferón gamma y células de memoria. Eso se hace en los cortes de 3, 6 y 12 meses. Hasta ahora hemos analizado, bueno, es más en realidad de 1.200 muestras, todas estas determinaciones, pero lo que les quería mostrar son unos resultados que ya hemos enviado a publicar a Cell Reports in Medicine, están en la última etapa de revisión, y es una porción de esta cohorte de 288 individuos vacunados con la vacuna Sputnik. Son todos personal de salud de la provincia de Buenos Aires, de estos 7 hospitales, y se dividieron 2 cohortes, de 18 a 59 años y mayor de 60. Y lo que medimos hasta ahora, como les decía, la medida de anticuerpos, anti-S, medida de anticuerpos neutralizantes, luego de la primera y luego de la segunda dosis de Sputnik, y recién ahora estamos en el punto de los 3 meses midiendo la inmunidad celular. Entonces, a medida que empezamos a analizar los resultados, rápidamente nos encontramos con algo que en ese momento nos sorprendió. Ahora ya nos sorprende porque después les voy a mostrar que hay un par de trabajos más publicados. Es lo que nos permitió dividir a los individuos vacunados en 2 grupos. En el grupo 1, de ahora en más lo voy a llamar grupo 1, son vacunados que llamamos naive, es decir, que en la muestra 0 no tienen título de anticuerpos, y cuando fuimos a corroborar estos pacientes, digamos, o estos individuos de qué se trataban, son individuos que no habían tenido COVID, no estaban registrados en la base de datos como diagnosticados con COVID. Y por otro lado, el resto de los vacunados, que llamamos preinmunizados, son aquellos que mostraron niveles de anticuerpos positivos antes de la vacuna, y estos individuos tenían todos COVID positivo por PCR. Entonces, este es el gráfico de los resultados del título de anticuerpo anti-spike. Fíjense, grupo 1, condición basal antes de la vacuna, tienen niveles basales o no detectables de anticuerpos, mientras que los del grupo 2 presentan anticuerpos en cantidades variables, como se ha demostrado en los distintos trabajos de fase 1, fase 2 de las distintas vacunas, que muchos individuos tienen niveles preexistentes de anticuerpos que pueden ser debidos a, digamos, a infecciones naturales por otro tipo de virus, pero cuando estos reciben la primera dosis, los del grupo 1, es decir, los individuos naive, nuevamente vemos una distribución muy heterogénea en el título de anticuerpos. Esto mismo se observa en distintos papers, mientras que los vacunados del grupo 2 que reciben la primera dosis, fíjense, muestran niveles de anticuerpos muy superiores a los individuos que no tenían títulos de anticuerpos al tiempo cero. Y aquí lo que se observa es un aumento del título de los niveles basales luego de la primera dosis de aproximadamente 20 veces. Es decir, que estos individuos convalecientes de COVID desarrollaron estos anticuerpos durante la infección, y aquí lo que se está haciendo con la primera dosis es intensificar la producción de esos anticuerpos, y vemos que desarrollan títulos muy altos de anticuerpos. Cuando se les da la segunda dosis a los vacunados del primer grupo, es decir, que no habían tenido COVID anteriormente, se observa que en un 100% aumentan el título de anticuerpos, aumenta pero mucho menos que los pacientes del grupo 2, mientras que estos del grupo 2 que reciben la segunda dosis, prácticamente el título no aumenta y se mantienen esos valores elevados. Entonces, luego de la primera dosis, lo que observamos es un 94% de cero conversión de estos pacientes. Si uno los divide en menores de 60 y mayores de 60, los números son importantes. La mayor parte de los que reciben la primera dosis hacen la seroconversión para menores de 60, el 89% para mayores de 60. Pero bueno, lo interesante que vemos es que, fíjense, si uno compara los títulos de los anticuerpos en aquellos del grupo 1, es decir, que no tuvieron COVID y reciben las dos dosis, este título es menor que los del grupo 2, es decir, los convalecientes que reciben una sola dosis y tienen títulos de anticuerpos mucho más altos que estos otros pacientes que recibieron las dos dosis. Cuando vamos a analizar los anticuerpos neutralizantes, se hicieron por dos métodos. En uno de los métodos se utiliza un pseudovirus que expresa GFP, virus de estomatitis vesicular, y en el otro método se utiliza directamente el virus nativo de SARS-CoV-2, y con los dos métodos vemos prácticamente lo mismo. Entonces, estos son los resultados para el grupo 1. Fíjense que los que no tuvieron COVID previamente, obviamente no van a tener anticuerpos neutralizantes. Cuando se reciben la primera dosis, desarrollan y en forma muy heterogénea niveles de anticuerpos o títulos de anticuerpos neutralizantes, y cuando reciben la segunda dosis se incrementa ese título de anticuerpos. Lo mismo se observa con este otro método, pero si uno lo compara con el grupo 2, fíjense que ya tienen en su condición basal anticuerpos neutralizantes en distintas cantidades. Esto es algo que se observa en muchos individuos que salen de COVID. Algunos tienen anticuerpos, otros no, pero es muy heterogéneo, por lo cual la respuesta inmune post-infección depende de cada individuo. Pero lo interesante es que cuando reciben la primera dosis de la vacuna, fíjense cómo aumenta marcadamente, más de 5 veces el título de anticuerpos, mientras que si reciben la segunda dosis, este título de anticuerpos se mantiene prácticamente constante, y lo mismo vimos por los dos métodos. No hay una diferencia significativa en la variación del título de anticuerpos si reciben una dosis o dos dosis los que previamente tuvieron COVID. Entonces, bueno, en este momento estos resultados a nosotros nos sorprendieron, pero luego aparecieron otros trabajos y demuestran prácticamente lo mismo con vacunas de la plataforma de RNA. Este es un trabajo donde se comparan de nuevo individuos que reciben, son 100 individuos, 88 reciben Pfizer y el resto Moderna, pero fíjense que se observa exactamente lo mismo. Hay algunos vacunados que tienen niveles basales de anticuerpos. Cuando reciben la primera dosis, en 4 o 5 días estos niveles ya aumentan en forma rápida, mientras que aquellos, y estos eran individuos que habían tenido COVID, mientras que los que no tienen COVID les demora un tiempo más extendido desarrollar anticuerpos, pero luego que reciben la primera dosis los niveles se mantienen constantes. Es decir, que aquellos que tuvieron COVID no solo aparece mayor cantidad de anticuerpos, sino que lo hacen más rápido luego de la primera dosis. Y en estos trabajos también lo que se muestra es que en estos vacunados que tuvieron COVID se observa una mayor intensidad, aunque no llegan a ser severos las reacciones adversas, solo las sistémicas, no las locales. Y el otro trabajo también es un trabajo que hay publicado para la vacuna de Pfizer, y muestra exactamente lo mismo que lo que vimos nosotros con la de Sputnik, es decir, individuos que estuvieron infectados previamente. Este es un trabajo con más de 1000 individuos, ellos mostraron 35 que tuvieron COVID, y exactamente se ve lo mismo. Tienen niveles basales previo a la vacunación, y luego de la vacunación aumenta marcadamente el título, pero no así entre la primera y la segunda dosis. Es decir, que los resultados que nosotros obtuvimos con la Sputnik en nuestra población son muy similares a los que se obtienen con estas otras vacunas en otras poblaciones, y estos son los primeros resultados que se van a publicar de Sputnik fuera de Rusia. Entonces, rápidamente, como conclusión, una dosis de Sputnik produce una alta seroconversión en individuos naive, mayor al 90%, aumenta el título muy marcadamente en individuos que tienen una inmunidad previa o que son convalecientes, que han sufrido la enfermedad. El título de anticuerpos anti-S y antineutralizantes en convalecientes es mayor que en individuos naive que recibieron las 2 dosis, y no se observó un aumento en el título de anticuerpos anti-S ni neutralizantes en convalecientes con la segunda dosis. Por lo tanto, en individuos que han tenido COVID, esta es la pregunta que ha ayudado aquí a tomar decisiones sanitarias, y ya estamos viendo que en otros países se están adoptando las mismas decisiones. Entonces, un individuo que tuvo COVID, ¿es necesario administrarle la segunda dosis? Hemos visto que la segunda dosis no cambia los títulos de anticuerpos totales ni de anticuerpos neutralizantes, por lo cual, como les decía, esto aquí todavía en Argentina no se hace, pero en otros países el que tuvo COVID en algunos países recibe solo una dosis de la vacuna. Y esto sí es importante y permitió adoptar políticas sanitarias a nivel de ministerio. Aquellos que han tenido COVID y reciben la primera dosis, tienen título de anticuerpos neutralizantes suficientes para donar plasma. Bueno, y esto es algo que se ha adoptado aquí en la provincia de Buenos Aires. Los que han recibido la primera dosis y han tenido COVID, obviamente que se realiza el análisis, pero pueden donar plasma de convaleciente para tratamiento de patologías. Entonces, estos resultados lo que muestran es que la infección natural hace de boost, y la primera dosis de la vacuna sería un prime, por lo cual lo que se está haciendo aquí con una única dosis es activar la memoria y generar o intensificar los títulos de anticuerpos neutralizantes, que sabemos que el virus es altamente sensible a los mismos. Bueno, eso es todo, muchas gracias, y dispuesto a contestar preguntas si es que las hay.
Bueno, mil gracias, Guille. Excelente, como siempre, tu presentación, y aprendimos mucho. Las preguntas van a ser al final, ¿sí? Voy a hacer mi presentación ahora. Voy a compartir la pantalla con ustedes. Bueno, ¿qué tal? Creo que me conocen. Soy la doctora Liliana Verosnik y yo quisiera contarles quizás otra cara sobre la respuesta a vacunas, que son los reportes de efectos adversos. Ustedes saben que la vacunación, conjunto de el agua potable en el mundo y también la penicilina, ha cambiado el contexto de la salud pública. Realmente fue revolucionario y la historia, a partir de estos eventos tan importantes para la salud, cambió realmente el ritmo de las infecciones. Aunque no nos quedemos exentos, ya nos mostró la pandemia actual, de nuevos desafíos. Pero cuando nosotros estamos frente a estas nuevas formas de poder intervenir en la salud, que son sumamente beneficiosas, también podemos tener algunos riesgos, que son los llamados famosos efectos adversos que están relacionados con las vacunas. Pero mi idea es, dado que es tan importante entender qué pasa frente a estas nuevas formas de inmunización, porque además de saber lo que mostró Guillermo, la importancia de que nuestra esperanza está en poder vacunarnos y que estas vacunas nos crean inmunidad y sobre todo, como vamos quizás a hablar después, al final o en la discusión sobre las nuevas variantes, la importancia que estas vacunas además sean seguras. Estos virus, este virus específicamente causó más de 120 millones de personas infectadas en más de 200 países. Y hoy, como les dije recién, es un gran desafío, por lo cual es muy importante comenzar a tener el efecto rebaño a través de las vacunas, porque este virus, que es un ARN, un ácido ribonucleico, tiene la capacidad de tratar de ir tratando de esquivar a nuestro sistema inmune y producir nuevas variantes cada vez más agresivas. Por lo cual es muy importante tratar hoy de tener vacunas que sean eficaces para tratar de reducir la infección. Y como contó Guillermo, ya sabemos que hay muchísimas plataformas hoy de vectores, de vacunas vectoriales, como está la vacuna Sputnik del Instituto Gamaleya, la vacuna AstraZeneca, la vacuna CanSino, que sabemos que estas vacunas hoy nos están dando realmente es total, o como la de Janssen, que realmente es un gran desafío y que es importante tenerlas, como las vacunas basadas también en ARN mensajero, como están las del Instituto Pfizer y la del Instituto Moderna y el Instituto de Enfermedades Infecciosas y Biotech, y las vacunas inactivas, como son las vacunas de Sinopharm y las Sinovac, y las nanopartículas, que son las nuevas vacunas, como las recombinantes con ayudantes de Novavax. Nosotros en la Argentina, perdón, pero estoy con un problema que no me deja pasar, perdón. No puedo pasar, perdón, No sé, voy a dejar de compartir y voy a volver a compartir, perdón. A ver, si me deja. Voy a necesitar un poco de ayuda. Perdón.
Sí.
Ahí está. Bueno, volvemos acá, que la importancia de poder garantizar una buena vacunación es demostrar a la población la importancia de estas vacunas que son confiables y que van a producir la menor cantidad de efectos adversos. Mi idea era demostrar un poco qué pasó con las vacunas dadas en la Argentina, dada que, como comentó Guillermo, la vacuna Sputnik 5 tiene menos publicaciones y es uno de los países pioneros en utilizarla, como los países de Asia y de Europa del Este. Nosotros tenemos un sistema integrado de información sanitaria que se denomina o está integrado por 3 grupos, que es el Sistema Integrado de Información Sanitaria, SISA, donde vemos reportar los efectos adversos que se producen frente a ciertas infecciones y ciertas vacunas. Esto lo recopila una comisión que van a estar, que es ESAVI, que van efectivamente a reportar a los eventos supuestamente atribuidos a la vacunación a una Comisión Nacional de Seguridad de Vacunas que se llama CONASEVA, que hoy tengo el honor de integrar. La realidad que la función del SAVI es poder entender si el efecto adverso está relacionado en sí con el producto, relacionado con un defecto en la calidad, con un evento coincidente que vamos a ver ahora que puede ser, o que puede estar relacionado con algún momento emocional, el estado ante la vacuna, o algunos momentos o algo coincidente que puede ser, por ejemplo, alguna enfermedad que este individuo traía. Por lo cual, cuando nosotros hablamos de reacción relacionada con el producto, es en sí con algún componente de la vacuna, con defectos en la calidad, que no es en sí algún componente de la vacuna, sino quizás, por ejemplo, algo que está relacionado con la producción o con los frascos. Es un ejemplo donde contienen las diferentes vacunas, errores de la vacunación que pueden ser causados por un defecto en la manipulación vacunando, o por un error programático, por ejemplo en la Sputnik, que nos ha pasado poner la segunda dosis en vez de la primera, o reacciones realizadas por la ansiedad frente a la vacunación, y los eventos coincidentes que no están relacionados en sí por la enfermedad o por la vacuna, sino por enfermedades que están concomitantes en ese momento, o por ejemplo accidentes. La realidad que nosotros hemos presentado ya en ESAVI más de 6.500% de notificaciones de 24 jurisdicciones de nuestro país, que fueron 49.301 eventos en estos 6 meses, de los cuales tenemos 641 eventos notificados como graves. Nosotros hablamos de eventos graves cuando estos eventos causan la muerte del paciente o ponen de riesgo la vida inminente, causan hospitalización o prolongan la estancia en aquel individuo que tiene que estar hospitalizado, produce una discapacidad significativa o produce en mujeres embarazadas en su producto una anomalía congénita. La realidad que las notificaciones que hemos tenido fue una media de edad entre 32,9 a 50 años, la edad fue de 40 años, con un predominio mayor en mujeres, con una demora de notificación de 4 días, la mayoría de las notificaciones fueron en la capital de la Argentina y en La Plata, de donde proviene el doctor, que es la capital de la provincia de Buenos Aires, el doctor Docena, de los cuales 1,3 de las notificaciones fueron eventos graves. La realidad, como estamos muy contentos, aunque por supuesto que quisiéramos tener más dosis de vacuna. En estos días, es decir, en el día de ayer, hemos tenido ya la suerte de poder vacunar a 18.870.000 personas, que el 7,93% de esas personas tienen ya 2 dosis. La realidad que fíjense que yo había preparado esta charla de Soy Real con un informe, que es nuestro, el 11º informe de vigilancia del grupo CONASEBA, presentado el 31 de mayo, tan solo hace 20 días, donde presentábamos qué había pasado en estos 6 meses, y fíjense que para el 23 de junio, es decir, que el día de ayer creo que nos queda ya viejo lo que voy a presentar, y prometemos que muy pronto vamos a poder compartir con ustedes los nuevos eventos. La realidad que desde el corte que voy a hacer del 9 de abril, de los eventos que fue de 5 millones sobre 5.493.153 dosis, podemos decir que tuvimos la mayoría, como contó el doctor Docena, de las vacunados con la Sputnik, seguidos por la Covishield y AstraZeneca, pero Sinopharma fue la mayor cantidad de vacunas que hemos podido instaurar. La realidad que los efectos fueron leves a moderados en un 90%, y 280 eventos, 0,95 requirieron hospitalización o fueron reportados como grave. La realidad que va a llevar un espacio mayor la vacuna Sputnik, porque fue realmente la primera vacuna que tuvimos la oportunidad de comenzar a inmunizar el 29 de diciembre del 2020, y lo que nosotros pudimos observar, que registramos 25.426 eventos relacionados con la vacuna, con una tasa de 740 en 100.000, pero que la mayoría de estos eran eventos sistémicos, pero no graves, como la fiebre, la cefalea, mialgia, altralgia, reacciones locales, diarreas, vómitos y náuseas. Y ahora voy a hablar un poquito de la anafilaxia y otros eventos que sí vale la pena contarles, pero que la mayoría de los eventos, como pasa con el resto de las vacunas, dura 48 horas y la recuperación de los pacientes es al íntegro. La realidad que hay muy pocas publicaciones, por eso nos pareció importante mostrar estas dos, donde hablan sobre todo la primera publicación del Lancet sobre Sputnik, de algunos efectos adversos, como es la mielitis transversa, de los cuales 3 pacientes se presentaron en este reporte, y de los cuales un solo paciente sí fue relacionado con vacuna y los otros eran coincidentes. Uno era indeterminado, y el otro era coincidente porque era el comienzo de una esclerodermia, la primera manifestación de ese paciente. Nosotros, en la Argentina, en el hospital italiano, se hizo un reporte también sobre 683 trabajadores sanitarios vacunados, que el 71,3% presentaron efectos adversos que no fueron importantes, sino que fueron leves y moderados, como les voy a mostrar en la tabla que sigue, y solamente el 0,1% requirió hospitalización. Esta es la tabla en la cual, como no quiero decir que, como van a ver, los efectos son los que ya les dije, pero sí tuvimos reportado 6 pacientes con anafilaxia, y lo importante fueron el reporte, que no están en otras publicaciones, y por eso estamos siguiendo muy atentamente, del síndrome de Guillain-Barré, que representa el 0,075, y también la trombocitopenia inmune. El resto de los eventos fueron errores programáticos o lipotimia por ansiedad a la vacunación, eventos coincidentes, tuvimos coincidentes muchos pacientes que estaban cursando COVID y se vacunaron, y bueno, realmente que los eventos secundarios fueron más que nada relacionados por la infección aguda que la vacunación, y sí, no concluyentes, 2 pacientes que presentaron, uno una pericarditis y un aborto en el primer trimestre del embarazo. Y hay 663 casos en análisis. Bueno, ¿cuál es nuestra experiencia sobre la vacuna AstraZeneca Covishield? Que sabemos que es eficaz. Aquí sí tenemos varias publicaciones en el mundo donde sabemos que previene la hospitalización y la admisión en unidades de cuidado intensivo y la muerte por COVID. Por lo cual es muy importante sacar los mitos sobre los efectos adversos sobre estas vacunas, sobre todo en nuestro país, y creo que también en los países vecinos. No sé lo que está pasando en Estados Unidos, que después me gustaría escucharlos, o en Australia, o en Europa, frente a el fantasma de la trombocitopenia, con la trombosis con trombocitopenia autoinmune. La realidad que nosotros entre las 2 vacunas, la Covishield y AstraZeneca, ustedes saben que es la misma, que hemos tenido más o menos 700.000 pacientes vacunados. Fíjese que no tenemos grandes efectos adversos registrados, sino los mismos que hemos obtenido, como la fiebre con cefalea o sin mialgia, artralgias, fiebre como único síntoma, la lipotimia, el síncope, y también concomitantemente, vuelvo a repetir, hemos tenido varios pacientes que cursaban COVID en el momento de la inmunización. La realidad que lo mismo nos pasó con la vacuna AstraZeneca. Nosotros tenemos la fabricación en este momento en la Argentina. Habíamos tenido un retraso por un problema en el envase de estas vacunas, por lo cual se retrasó, y ahora creo que este estado, es decir, que esto se va a reponer, por lo cual estamos vacunando en forma realmente muy importante a los pacientes en la Argentina. Pero ¿qué es lo que hemos visto? O lo que está en base a estos eventos poco frecuentes. La famosa trombocitopenia autoinmune inducida por heparina. El primer grupo lo reportó el doctor Greinicher, donde él muestra luego de la administración de 200 millones de dosis este evento, que también es producido por la vacuna de Janssen, y que fue este evento reportado luego de la administración de 6.800.000 dosis. La trombocitopenia autoinmune todavía no es muy claro cuál es el mecanismo o el por qué se produce, mientras que saben que algunos de los pacientes se presentan como cuadro subjetivo de coagulación intravascular diseminada, en otros realmente presentan plaquetopenia, que se identificó como un mecanismo inmunológico que induce una disminución de la plaqueta inducida por heparina, y que se produce por una alteración del factor 4, y esto no hay riesgo con factores de riesgo asociado a este evento, como vamos a ver. Por lo cual, tanto la Agencia Europea de Medicamentos EMA como la Agencia Reguladora de Medicamentos y con otros productos sanitarios del Reino Unido afirmaron que esto es un evento sumamente raro, que la realidad, por lo cual en este momento creen que todavía es una vacuna que se es aconsejable y hay que seguir administrándola. La realidad que esto me parece muy importante recalcar. La trombosis por SARS-CoV-2 es un evento sumamente frecuente. En infecciones naturales hemos tenido reportes de 7,8% en embolia pulmonar, 11,2% de trombosis venosas profundas, 23% de tromboembolismo venoso, 1,6% de accidentes cerebrovasculares y 30% de trombocitopenia registrados quizás por COVID-2, por la infección natural. Pero además, la trombosis no es un hecho no frecuente en la población general. Fíjense que existe en la Unión Europea un registro por año de 100.000 personas que desarrollan trombosis, que alrededor de 3.000 casos por mes desarrollaron efectos de trombosis en Reino Unido por diferentes causas. Mujeres ante consumo de anticonceptivos se registra una en 2000, y la trombosis con trombocitopenia causada por la vacunación es de 0,0004%. Creo que estos son los números que nos deben convencer para ayudar como médicos, porque hay muchos médicos que también no están convencidos, por eso me pareció muy importante demostrar la importancia de la vacunación y la poca frecuencia de estos eventos. Por lo cual, tanto el grupo GAPS, que es el Grupo Asesor Global de Seguridad en Vacunas de la Organización Mundial de la Salud, afirman que los eventos bajo evaluación de que es la trombocitopenia autoinmune inducida por heparina, son muy raros. Por lo cual, en nuestro ministerio siguió aportando que debemos vacunar y seguimos vacunando, y esto sí es un dato actual que todavía no lo hemos publicado, pero se está por publicar, donde sobre 1.456.714 dosis con AstraZeneca y Covishield no se ha identificado hasta ahora los eventos según característica de del grupo de Victor Collaboration. Es decir, no hemos visto trombocitopenia autoinmune inducida por heparina. Por lo cual, en este informe que va a salir en esta semana, donde se recalca la seguridad de la vacuna, especialmente AstraZeneca, y nuestras recomendaciones. ¿Puede administrarse la vacuna en pacientes con antecedentes de trombosis? Sí. Las personas con antecedentes de trombosis tienen un riesgo mayor de volver a presentar una trombosis, pero no con la vacuna. Sí tiene un riesgo muy elevado con la infección natural por SARS-CoV-2. ¿Puede administrarse la vacuna en pacientes con riesgo de trombosis o trombofilia? Sí. Los pacientes con antecedentes de trombosis u otras condiciones de riesgo conocidas como factor 5 de Leiden o mutaciones protrombóticas que producen trombofilia o la trombofilia en sí, se benefician con la administración de la vacuna por el riesgo de presentar cuadros de trombosis ante la infección natural. Por lo cual es muy importante decir que no hay riesgo. Y lo otro es que las personas anticoaguladas también pueden recibir las vacunas. Sabemos que las vacunas son en forma intramuscular, pero dada por personal entrenado, son sumamente seguras, por lo cual no es una contraindicación tampoco las vacunas en personas anticoaguladas. Y ahora sí, pasamos a las otras vacunas dadas en mi país, en la Sinopharma, aplicadas en 1.295.940 personas, tuvo un riesgo de 31,6% dosis de producir realmente fiebre con cefalea, cefalea con mialgias, anafilaxia, se vio en un caso, que creo que esto es muy importante, y síntomas gastrointestinales como sabor metálico en la boca. Hubo errores programáticos en personas que recibió como segunda dosis otra vacuna o que habían recibido inmediatamente la administración de plasma, y por supuesto coincidentes fueron infecciones coincidentes como les pasó 46 pacientes que estaban cursando SARS-CoV-2, es decir, la infección natural, fueron inmunizadas y realmente los efectos graves y hospitalizaciones fueron por la infección natural, no por la vacuna, y tenemos 31 casos que están en análisis y datos que estamos todavía, 2 pacientes que no tienen datos para ser analizados. Quiero mostrarles el reporte también internacional de efectos trombóticos relacionados con la vacuna. Fíjense que en todo el mundo se han reportado, y sobre todo con la vacuna de Janssen, 15 notificaciones en España, en Canadá, en Reino Unido, es donde más se ha visto estos efectos trombóticos, pero también en Francia, que tienen un alto número. Pero esto sigue siendo realmente una tasa baja contra el beneficio que es vacunar. Y fíjense, esto es el reporte de seguridad de las vacunas frente a los eventos graves o no graves. En Estados Unidos, la vacuna Pfizer ha mostrado sobre la notificación de 9% de efectos graves y de 91% de no graves. La de Moderna, sobre una notificación de 200 por 100.000, de 3% de casos graves, 97 de no graves. En España, la mayoría fueron no graves, lo mismo con Pfizer, Modernas y, bueno, AstraZeneca, que es la Vaxeria. En Canadá, con las 3 vacunas también, pero ellos reportaron varios efectos graves en los pacientes, 14,9%, creo que es el país seguido también con Francia de estos eventos. El Reino Unido, la mayoría de ellos, los reportes fueron no graves, aunque tuvieron, creo que la mayor cantidad de efectos por trombosis inducidos por plaquetopenia y por factor 4, es decir, la forma autoinmune reportada. Y en Francia sí han reportado un 22% con la Moderna, 16% con la Pfizer, y con la Oxford AstraZeneca un 27%. Por eso me pareció muy importante mostrar los efectos adversos y la seguridad en mi país. Bueno, nosotros sabemos que las indicaciones para las personas que han recibido la vacuna, si presentan síntomas, uno tiene que tener claro que esto puede estar relacionado por la vacuna, pero si estos síntomas son no habituales y persisten más de 48 horas, deben ser reportados. Y como médicos debemos nosotros saber que en mi país se debe notificar al CISA y el CISA al SABI. Y esto debe ser nuevamente reportado con historia clínica, con todos los datos precisos de lo que ha pasado el paciente, porque es muy importante, porque no solamente va en la salud del paciente, sino en la seguridad frente a la vacuna y a nuestra experiencia para poder saber qué hacemos o si seguimos prescribiendo ese tipo de vacunas. Por ahora tenemos la suerte de decir que los efectos adversos son menores. Sabemos que nos presentamos frente a un momento muy crítico, porque esta primera infección que se transformó en pandemia nos lleva a recapitular qué va a pasar, porque hemos ya tenido otras infecciones que fueron epidemias sumamente importantes en Asia. Hemos sufrido una pandemia, pero que rápidamente pudimos controlar, como fue la influencia estacional o la influencia porcina, pero sabemos que por año 290.000 a 650.000 muertes son causadas por infecciones respiratorias, por lo cual frente también al desafío de las nuevas variantes del COVID que producen aumento de la transmisibilidad, aumento de la virulencia, y estamos pensando qué pasa con la eficacia de las vacunas, por lo cual es producen medidas nuevamente de estrés en la sanidad. Qué suerte de poder contar con vacunas que sean seguras, porque sigue siendo nuestra esperanza, por lo cual sí creo que podemos seguir utilizando en mi país, y creo que también han demostrado ser las vacunas seguras en el resto del mundo. E invitarlos a nuestro congreso, y muchísimas gracias por su tiempo.
Hola, buenas noches. Hello, good evening. I am Antonio Condino, member of LAZIT. I would like to thank Liliana and Ekaterina for this kind invitation to present on some data about the vaccination against COVID in Brazil. So I would like to share my screen. So I will be brief because many points of this talk were already covered. So a first concept is about herd immunity. And this we are going to reach, to accomplish in about over 75% of people immunized. This is critical, especially for our primary immunodeficient patients, because depending on the immunological defect, we are sure that they will be unable to produce antibodies. We are not sure about cellular mediated immunity. And eventually, there are those kind of patients that will have a partial response, antibody response, and some of them will respond or not. So vaccinating the general population will eventually provide safety for them. It's a group protection. The coronavirus, as we already have seen, is the principal component that the vaccines are directed against currently is they are directed to stimulate antibody production against the S protein. So here in Brazil, we are using the Pfizer vaccine, which is a genetic vaccine with over 97% efficacy. We are using the Oxford AstraZeneca vaccine, which is a vector vaccine, and will receive very soon the Johnson vaccine. So they are produced over adenovirus platform that will receive the sequences of the S protein, and they are inactivated. That said, safe for our patients. And we have the Sinovac, the CoronaVac vaccine, which is based on the whole inactivated virus. This vaccine has around 50% efficacy, so that means that it reduces the chance in 50% of the person to get infected. However, this vaccine has been proven to protect in a very good way, very efficient way, the severe forms of the disease. And here in our country, we are already detecting this kind of positive effects. So these are today numbers in Brazil. I pulled this from Folha de São Paulo newspaper. So what we have 41% of our population already immunized with the first dose, about 15 receiving the second dose, and these are the total numbers. We have 18 million people here and unfortunately 500,000 people dead in our country. About some statistics. So these are the numbers. We are now second place in the world after the United States, and this is the statistics, the relative statistics, the mortality per 100,000 inhabitants. We have an intermediate number. So what happens here is that our country is very populous and very big. I would like to share another screen with you to show some actual numbers here. So this is the number, 68, almost 67 million people already received the first dose of the vaccines. That represents 42.5% of our population, and about 25 million people, which represents 15% of our population already received the second dose. Our state, São Paulo, is more or less on the same proportion. Mato Grosso state is the leader in our country for performing the immunization. We have to consider that this state is less populated compared to the other Brazilian states. This is the total number statistics. São Paulo is the most populous state in Brazil, so has the population similar in number to Argentina, for example, and we already gave the first dose to more or less 50% of the people. So what I want to show you here, in one side, we are going relatively, I wouldn't say that much slowly, but we are delayed in our vaccination program compared to other countries that started earlier than us. And the reason is the behavior of the federal government that decided to adopt chloroquine and ivermectin as an early treatment, and did not buy the vaccines in the last year, and decided just to do it now in the last time. So let's have a comparison with the world. So this is the world map. In terms of number, this is our statistics. So our index is about 430 doses per 1,000 inhabitants. And here is our population. So Brazil is a continental country and very populous. And it is, I would say, it's really a challenge to perform all that vaccine. So the effort, I would say, to compare is more or less like the United States. However, the United States has a more powerful economy and started much earlier. So although the former president of the United States behaved on the similar way of our current president, anyway, the institutions worked on a positive way, and the United States is performing a good job in terms of vaccination. Afterwards, I would like to listen the comments of our colleague, Kate Sullivan, because she lives there and she will have more details. But I would say that for now, we are accelerating the vaccination rate. Just last week, Brazil was the #1 in the world to give the number of doses on a single day. So we are doing the best as we can to overcome this situation. And we have to keep confident that we will control. Actually, I would say that we are currently facing a threat of the third wave to affect us, but apparently is not that bad compared to the first and second wave. So apparently the vaccines already started to protect us, to work on a positive way. And additionally, the death profile is now more focused on people of middle age. So the old people in Brazil stopped to die, and this already reflects the positive effect of the vaccines. So there is hope in this way. If I would be the Brazilian Minister of Health, so the first thing I would do is to stop the bullshit speech as our president currently makes, because his speech is much uglier than what the real numbers and the real efforts reflect the country. So we are actually much better than our presidents. Unfortunately, Fiocruz and Butantan are working very well and as much as they can to help us control these pandemics. So as I said, I would like to be brief because most of the explanations about the vaccines were already presented by Guillermo and Liliana, and I will stop here, and afterwards I will be very happy to answer questions. Thank you.
Alfonso, el video de, please, the record of Dr. Stewart.
Okay, good morning, or good afternoon, good evening. Welcome to the LACID COVID seminar webinar. Thanks for the opportunity for inviting me to be involved with this. I'm coming to you from Sydney this morning, and I hope you're all doing well. Thanks to Liliana and everyone in LACID for inviting me to be part of this special event. So as you all know, we are in the ongoing grip of a global pandemic. This is all about SARS-CoV-2 and COVID-19. As of a few days ago, according to the WHO, there'd been nearly 180 million cases of COVID-19 across the world, and tragically approaching 4 million deaths, which is just a dreadful statistic. Optimistically, though, the numbers of vaccines doses that are being rolled out is increasing literally by the day. So not nowhere near what we need, but at least that is certainly moving in the right direction. So this just shows how SARS-CoV-2 COVID-19 has really ravaged the world over the last 18 months. I don't think any of us have lived through anything like this ever. And this just shows this is starting in January 2020, and this is up to present day. You can see the number of cases that have come through various countries, various continents, various communities, and so on. And it really shows it doesn't matter where you are. COVID-19, either the wild type strain or now being grouped with all the variants of concern, particularly the Delta strain, is really causing havoc everywhere. So you can see countries like the US have just had this wave that's gone on and up and finally coming down now. France, likewise, up and down, several waves and the like. So no country has been immune, sorry about that pun, immune from the effects of COVID-19 and SARS-CoV-2. So how deadly is SARS-CoV-2 infection? Well, this shows the case fatality rate for many, many different countries. And this is actually the absolute number of deaths in those countries. So something like the US, which has had 500,000 deaths or thereabouts, versus Australia, where I'm coming from, has had just under 1,000 deaths. But you can see the case fatality rate, it ranges, it's between that sort of 1 and 5%. Globally, it's about 2.2%. So about 1 in 40, 1 in 50 people who get SARS-CoV-2 infection are tragically dying. But it sits in that 1 to 5% band, generally speaking. I've listed some of the Latin American countries here for comparison. Australia, India, the US, and so on. A few outliers. Mexico is quite high. Yemen is very, very high. But generally, it's pretty tracking very similarly everywhere. So we knew from the very beginning of the pandemic that there was a number of risk factors for SARS-CoV-2 infection and mortality. The first one that became very obvious was age. So even though the proportions of people who were getting infected didn't really differ that much across each decade, except for very young children, the death rate really shifted according to age. So with every decade of age, the incidence of death increased. So I actually turned 50 last year. So I transitioned from this risk group to this one. So I'm now 3 times more likely to die of SARS-CoV-2 than I was 12 months ago. But you can see this stepwise increase every decade of life, becoming much more vulnerable to SARS-CoV-2 infection. And likewise, pre-existing conditions, health conditions, heart disease, lung disease, diabetes, cancer, and so on, were all risk factors, comorbidities for SARS-CoV-2 infection and COVID-19. But what about people with inborn errors of immunity? You'd think that people who intrinsically have a defect in their immune system due to monogenic variants that compromise a particular pathway or the generation of a particular cell type, excuse me, may be at increased risk of SARS-CoV-2 infection or disease in the context of their pre-existing inborn error of immunity, whether it be immunodeficiency or immune dysregulation. So that was a question that that I was involved in a large team of people last year asking, and this was really built on a couple of case reports that were appearing in the literature very early on in the pandemic, reporting variable disease courses in people who had different types of immunodeficiencies. So the first couple of papers came from Ana Rosa Sorencia and Isabella Quinty, who were looking at XLA patients, B-cell deficient patients. And surprisingly, they found that those individuals were actually fine, relatively speaking. They had very mild disease, and as these titles suggest, recovered quite okay. Then there's a couple of more cases that came out with BTK mutations, XLA, B-cell deficiency, and overall they were relatively mild. Then there was a few more specific cases. So there was a report of very recent of a patient with the STAT1 gain of function, a patient with X-linked SCID, patient with interferon gamma receptor, patient with ARP-C1b mutations, and then NF-kappa B2 haploinsufficiency. So there's a number of case reports that were emerging in literature over the last 18 months or so. So at the same time, or inspired by these as well, myself and many other people, particularly Isabel Metz, Klaus Vornatz, Kate Sullivan, Nazar Maloui, and so on, started thinking about this at a global level, trying to get a snapshot of what SARS-CoV-2 is doing in the context of people with inborn errors of immunity. So we ran, this was again done with ESID and the iOS committee and many other societies. LACET, of course, was part of this. Sent out a survey to clinicians trying to gauge what's the impact of SARS-CoV-2 on patients with inborn errors. So we ended up getting clinical information from 94 patients. They were very much from across the world, none from Australia, fortunately, but many from Latin America, from Europe, from North America, from the UK. The death mortality was about 10%, and I'll go through this in a little bit more detail in a moment. So that was our first glimpse of what SARS-CoV-2 is doing to the PID community. And since then, or along with that paper, several other sort of cohort studies were published coming from specific countries. So, for example, this paper is out of Iran, this study is from Mexico, this study is from the UK, this study is from Israel. And we've also had much more focused studies, cohort studies, a couple of studies straight out of New York, for example, looking at patients with primary immunodeficiency or CVID. And interestingly, the mortality rate from these different studies varies dramatically. So we reported about 10% mortality. That was confirmed by some other studies, but other studies found it was much higher. So, for example, this study out of Mexico was about double that, 20%. Likewise in the UK. Interestingly, the study in Israel, they had no mortality at all. So 20 patients, no deaths. But then Iran, very high. So it sort of depended on the location, the study, the types of diseases they had. But collectively, we're starting getting a picture of what SARS-CoV-2 infection was doing for patients with inborn errors. So from these 7 or 8 studies, just looking at the numbers, there's about 260 patients with a vast array of inborn errors, and the mortality rate is about 13%. Now, of course, that's much higher than what I showed you before for the global level. But just bear in mind, when we did this last year, the mortality rate was actually much higher, particularly in parts of Europe, in the US, where it was in that 5 to 10%. But overall, 13% mortality rate based on the numbers that keep appearing in the literature reporting in patients with inborn errors and SARS-CoV-2 infection. So I'm just going to talk a little bit about the study that I was involved in to give you a bit more specific information about that study. And I realize this is probably a bit of an unconventional way to show the data, but I actually like the way this is presented. This is also, I should acknowledge, Georgia Bicoli, who worked with Isabel Metz, who did a lot of the clinical analysis and so on. So this is a breakdown of our cohort. Most patients, over half of them, had an antibody deficiency. And then there were a number of patients who had combined immunodeficiencies, phagocytic defects, immune dysregulation, and so on. But the vast majority were antibody defects, so CVID, XLA, other types of antibody deficiencies. So this is broken up into the type of inborn error by color, and then by age around the wheel here. So we've got youngsters under 18, 18 to 55, and then over 55. And then each age group is broken down as to whether they had or did not have comorbidities, and then whether they needed to be admitted to hospital or not. And if they were admitted to hospital, if they were on ICU, it needed ICU or needed some type of ventilation. So the first thing you can see is many of the patients who succumbed to SARS-CoV-2 infection had antibody deficiencies. Many of them were in intensive care. So as shown here, for example, many of them were in the older age group, over 55. So 4 out of the 9 deaths that we noticed were in this over 55 age group, and most of them had comorbidities. So it was a little bit like what we saw in the general population, the older patients who had pre-existing health conditions. Interestingly, there were no deaths in patients with combined immunodeficiencies. So these are conditions like ZAP70 deficiency and some other monogenic defects that affect T cell function. And again, over here, when you look at some of these other types of inborn errors, the patients who did succumb were in ICU and often had comorbidities. So whilst the numbers were slightly higher in terms of mortality, the predisposing factors were pretty similar to the general population. So that was an interesting snapshot of what was going on. I must say the ages, because we were identifying younger patients than in the general population, the numbers of patients that needed ICU admission and the age range was much lower, higher for ICU admission, lower for age than the general population. But what was really interesting is sort of the information we could glean from this cohort. Now, whilst 94 is a pretty good number, as I said, there was lots of different types of gene defects or causes of disease amongst those numbers, but we could make some very broad generalizations. About 30% of patients were asymptomatic. They were picked up to be SARS-CoV-2 positive for incidental reasons, like they were traveling or they were needed to go to hospital for surgery or something like this, unrelated to their inborn error. They were tested and found to be SARS-CoV-2 positive, but actually asymptomatic. So about 30% of patients were asymptomatic. or mild disease. And many patients actually had, some of the patients that had mild disease were found to have autoinflammatory conditions. So a Cardi-Gutierrez syndrome, for example, FMA, familial Mediterranean fever was another one. Patients who had XLA or B cell deficiency had mild disease. Some of the combined immunodeficiencies, as I said, CGD, and patients who had impaired IL-6 signaling, so autosomal dominant hyper IgE syndrome due to STAT3 mutations, autosomal recessive hyper IgE due to PGM3 mutations, and STAT1 gain of function. They all had mild disease. The more severe diseases included impaired NF-kappa B signaling, interferon gamma receptor, and as I said here, some of the older CVRD patients who had those comorbidities. So thinking of that data gave us an opportunity to think more about the consequences of SARS-CoV-2 infection, what immune determinants might be driving some of the pathology, and how some of the therapies that are being used could be interpreted. So the first one that struck us was the IL-6 signaling. So as I said, we had a couple of patients. One had STAT3 loss of function. We also had another patient with STAT1 gain of function, which has a lot of clinical and cellular features like STAT3 dominant negative hyper IgE syndrome patients. And we also had a patient with PGM3 deficiency, this group of patients who have been shown to have impaired signaling through the IL-6 receptor. So arguably, that may suggest that STAT3 signaling, probably IL-6 signaling, drives that cytokine storm that we're so familiar with, and hence the desire to test tocilizumab as a therapy for SARS-CoV-2. But this probably provides some explanation as the mechanism of immune pathology in the context of IL-6, STAT3 signaling driving some of that cytokine storm. And if you can't signal through IL-6 for mutations that affect that pathway, or because the variant, in this case PGM3, prevents expression of GP130, you might be a little bit protected from disease. Again, I touched on this before, BTK mutations, XLA. Even before we did this work, there were studies that were being published on the use of BTK inhibitors like ibrutinib and others to treat SARS-CoV-2 in the general population. There were studies that were looking at SARS-CoV-2 in patients who were on BTK inhibitors because they had B-cell malignancies, or there were trials that were ran to look at the effects of BTK inhibition. And this did show some promise. And I think the interpretation of this is, excuse me, it's not because BTK inhibitors are targeting B cells, it's probably targeting monocytes, myeloid cells, which are making that IL-6. And it has been shown that BTK is important downstream of TLR ligand interactions to drive cytokine production by myeloid cells. And that's sort of shown here. What this is looking at is the level of phospho-BTK in healthy donors, and then in SARS-CoV-2 infected individuals, showing that BTK is activated in their monocytes, but not in their B cells. It shows again that the monocytes that are either resting or stimulated, their production of IL-6 is increased in COVID-19 patients compared to healthy donors. But then this doesn't happen in B cells. So the monocytes do make a lot of IL-6. So BTK inhibition is probably attenuating this somewhat. So there's probably good evidence there for why there's mild disease in some of those conditions. A lot of information on this, but this is just something else that wasn't really a finding from our study, but I'll come to the significance of this in a moment. Other aspects of SARS-CoV-2 in the general population, these 3 studies, one in PNAS, one in Nature Medicine, one in Science Immunology, have all shown that dramatic activation of the complement pathway is associated with severe COVID-19. So this Science Immunology paper shows this is otherwise healthy individuals, not PID patients, those who are in ICU or needed invasive mechanical ventilation. Their levels of C5a, for example, were much, much higher than COVID individuals who didn't need ICU admission or ventilation. And likewise, these are individuals who are uninfected or then who had low, medium, and high levels of viral infection, looking at all these complement factors. So on the left-hand side here, you can see there's this almost dose-dependent increase in these complement mediators. And over here, these regulators of complement decline with disease severity or level of infection. And the reason I'm showing you this is because there was this paper that looked at hereditary angioedema, which is a disease characterized by impaired complement activation. It was a small number of patients, 13 patients, but there was no deaths. So again, suggesting that whilst you may think a defect in a major pathway, whether it be IL-6, whether it be BTK, whether it be complement, may be a problem, those conditions may actually protect patients from severe disease. So what else can I tell you about inborn errors? So many of you would be familiar, and many of you are probably part of the COVID human genetic effort, which was set up by Jean-Laurent Casanova and Helen Su in the US. At the beginning of the pandemic, they brought together a global team of researchers, clinicians, DNA sequencing hubs, infectious disease specialists, with the mission to understand the genetics of severe COVID-19, or actually, on the other hand, the genetics of resistance to SARS-CoV-2 infection. So really trying to understand what genes influence our immune system and how we as individuals respond to SARS-CoV-2 infection. So their hypothesis is based on previous studies actually done by the Casanova lab and by Helen Su's team at NIH, where they, plus other groups, of course, had identified variants in genes in the type 1 interferon signaling pathway were actually causal or pathogenic in the context of severe infections with some other viruses. So, for example, herpes simplex virus, we know, is really prominent in individuals who have mutations that affect the TLR3 type 1 interferon signaling pathway. Individuals who have really bad problems with live attenuated viruses have compromised signaling through type 1 interferon type 1 interferon STAT2 signaling pathway. Influenza A virus, likewise, is impaired in people with IRF7, IRF9 mutations. And then other infections like rhinovirus and so on can be impaired by MDA5 mutation. So there's this suggestion that there's likely going to be people in the general population who have variants in this pathway who will be susceptible to SARS-CoV-2, but because this was a novel pathogen, never been seen before, we just didn't know who those people were. So to cut a long story short, the COVID human genetic effort published 2 papers in Science last year, about September last year, indeed showing a really important role for type 1 interferon responses in protecting the host against severe COVID-19. So the first study on the left here identified genetic defects in the type 1 interferon signaling pathway. So some of those genes I just mentioned then were found to be mutated in about 3.5% of individuals with severe COVID-19. So mutations in the components that are important for producing type 1 interferons or responding to type 1 interferons. But perhaps what was more striking was this paper by the same teams who found not gene defects affecting the interferon pathway, but actually found that individuals produced autoantibodies that neutralized endogenous type 1 interferons. So instead of having a genetic defect that compromised signaling through type 1 interferons, there was 10% of individuals, interestingly mostly males, who produced autoantibodies to type 1 interferons, and that actually blocked the protective effect of these cytokines, thereby both studies showing this important role for type 1 interferon signaling in host defense against SARS-CoV-2. So this is interesting because one of the patients that was in our study actually had GATA2 deficiency. And a feature of GATA2 deficiency, of course, is a lack of plasmacytodendritic cells or interferon-producing myeloid cells. And we would speculate wildly, but speculate that the cause of severe disease in GATA2 deficiency due to SARS-CoV-2 infection results from a lack of interferon-producing cells and that impaired innate immunity in those individuals. So continuing with the interferon pathway and studies, one of the early papers in this space reported that a patient who had APS1, so APOSED, air mutations, actually had very severe pneumonia. This is a patient from Italy, which was interesting because, as many of you would know, a feature of APOSED, apart from autoimmune disease and candidiasis, is actually the production of high levels of antibodies to cytokines. And one of those cytokines are the type 1 interferon cytokines. So this was followed up very recently by this study, again, that involved Jean-Laurent Casanova, Helen Su, but also in this case, Mikhail Leonakis at NIH. And they had a cohort of 18, I think, APOSED patients. And this shows that they all have antibodies against all the type 1 interferons, except for kappa and most epsilon. There's a few healthy donors on the side here. So they studied this cohort of patients to see what would happen to them, and they all had COVID-19. And what was striking was that they had, I think they must have had more, they had 22 patients, I think. 19 of the patients were hospitalized, which is a very high hospital admission rate. 68 needed ICU, again, which is very high, and 4 of them died. Now, of course, small numbers, but that's a pretty high death rate. Again, just reinforcing this role for type 1 interferon in protection against SARS-CoV-2. So if you have a mutation in the pathway or autoantibody, you are predisposed to severe COVID-19. And this, of course, has led to a couple of experimental efforts to test the efficacy of interferon therapy as a treatment for SARS-CoV-2. So this is a nice review written by Isabel Metz and colleagues on behalf of the COVID human genetic effort that was just published in Journal of Clinical Immunology. That's a review article. But these 2 papers here actually did try interferon alpha or interferon beta therapy in a couple of patients with inborn errors. These individuals had mutations in, as it says here, TLR3 and IRF3, and this individual had mutations in NEMO. There was, of course, they're not controlled studies, but there was anecdotal evidence that the outcomes of these individuals treated with interferon was much better than generally people who were not treated. So again, reinforcing the role of interferon. So I'm just going to spend the next couple of slides and finish my talk just talking about what do we know about actual immune responses to SARS-CoV-2 in people with inborn errors of immunity. So this is from a nice review from Alexandra Sete and Shane Crotty from the La Jolla Institute of Allergy and Immunology in California. And this is like textbook cartoons showing that early on your innate immune response responds, and over time your adaptive immune response kicks in the face of a generic infection. Antibodies come on, CD4, CD8 T cells respond. In SARS-CoV-2 infection, in the general population, people who have mild disease and cover, who have this lag time of when viral load is increasing over time, declines eventually, your innate cells do something, and then your adaptive cells, again, start making T cells and responsive B cells and antibodies, and things go okay from there. But though with severe SARS-CoV-2, viral persists, T cell dysfunction seems to be not so, T cell dysfunction seems to be prominent, antibodies are variable, and innate immunity in the context of like a cytokine storm keeps persisting. And this is showing it a little bit more detail here. This is, again, models of what we think the immune response in healthy donors to SARS-CoV-2 looks like. So this is your average COVID response. It's most of the viruses in your upper respiratory tract. Some gets into the lungs. In fatal COVID-19, the virus sort of goes everywhere and is very problematic in your lungs, obviously. The T cell response is predominantly CD4 T cells, but there's a contribution by CD8 T cells. And your antibodies, you have antibodies of different specificities, of course, but the most important antibodies are those against the spike protein, and of course, those that are neutralizing. So there's one study came out very, very recently from Mike Keller, who's in DC, and this actually studied antibody and T cell responses in SARS-CoV-2 patients who had antibody deficiency. So they studied 5 individuals from one family. Several of these individuals, the ones shown in black, actually were SARS-CoV-2. The patients shown here, P5, and a couple of the other ones were found to be SARS-CoV-2 positive, but those in the black circles actually had pre-existing specific antibody deficiency, hypogamma, or CVID. And they also looked at 2 other CVID patients unrelated to this family, and then were able to track their immune response following SARS-CoV-2 infection. So the top here, this shows antibody responses, and here's the antibody deficient individuals. So you can see here P1, P2, P4, and P6 are antibody deficient. P6 and P7 are the CVID patients unrelated. And then these are the immunocompetent individuals, 3 and 5 there from this kindred over here. So basically looking at antibodies against the nucleocapsid or spike, this is what the controls look like. From these 5 individuals here, except for this P6 who was hypogamma, they could detect antibody responses to the nucleocapsid as well as to the spike protein, which were not too dissimilar to levels in the immunocompetent controls. And then this is looking at CD4 T cell responses, again, in responding to different peptides corresponding to the membrane, envelope, nucleocapsid, or spike proteins of SARS-CoV-2. And again, for the immunocompetent individuals shown in the red, compared to the antibody deficient individuals, pretty similar responses, no significant differences. And in fact, there was this trend for higher responses in the antibody deficient individuals here. So at least in the context of natural viral infection, with the exception of this one patient who was A-gamma or hypogammaglobulinemic to start with, at least the short-term response was pretty intact. So as the authors concluded here, this shows robust T cell activity and antibody responses against SARS-CoV-2 in patients with antibody deficiency, generally speaking. But what about vaccination? We're hopefully moving into that phase where we'll see many fewer cases because the vaccine is being rolled. As I said at the beginning, billions of doses have been distributed. So this is a really nice study that just came out as well in Journal of Allergy and Clinical Immunology from David Hagen in Israel. They were lucky enough to study 26 patients with inborn errors, and they looked at these patients 2 weeks following their second dose of the mRNA Pfizer SARS-CoV-2 vaccine. And again, like some of those studies that looked at SARS-CoV-2 in inborn errors patients, this is a mixed array of mutations that cause these conditions. So this is first looking at antibody levels. So there's a lot of data here, but I'll just go through it. These are uninfected healthy donors, so people who have never seen SARS-CoV-2 before, so they don't make an antibody response. These are patients with inborn errors of immunity, and you can see that it's quite variable, and I'll come back to that in a second. These are inborn errors, patients who have been vaccinated, I should say. These are healthy donors who have been vaccinated, and these are people who had SARS-CoV-2 but are now convalescent. So comparing this color and the yellow symbols, you can see there's pretty good responses, slightly higher for the healthy vaccinees. But what's important to note here, and I should say this is for viral inhibition as well. So again, there was a range of responses in the patients, but generally speaking, it was much higher than uninfected individuals in the healthy population. But key point here is that 4 of the patients had XLA, and 1 patient had been treated with rituximab. So they were B-cell deficient, and variably antibodies were not being able to be produced because they had no B cells. So overall, when you take those patients out, 81% of their cohort, 18 out of 22, were found to make antibodies against SARS-CoV-2 vaccine. There's a bit more information here just showing there was good correlation between the levels of IgG produced against SARS-CoV-2 and the neutralizing capacity. And when you break it down into the different types of inborn errors, again, these are the patients who have no B cells, they're B cell negative, so they don't make a response, obviously. These are patients who have like autoimmune diseases, so these were individuals who had ALPS, for example. Complement deficiency response was totally intact. And then there was a bit of a difference between CVID patients, depending on age. So those under 50 generally had a better response than those over 50. And this is one patient with an inborn error who was infected, but then recovered. So basically, when you look at it in that context, at least in the short term, the antibody responses were encouraging, to say the least. They also looked at T cell responses, and this is looking at cytokine secreting cells in vitro in response to a whole range of peptides. And again, same layout here. These are before exposure, and then these are the patients. These are healthy vaccinees, and these are convalescent individuals. So again, in response to the vaccine, most of the patients could produce a T cell response. It was actually 73%. And then again, when you look at it, breaking it down, whether these are patients with inborn errors who do or do not have B cells, those who don't have B cells were capable of making a T cell response, which was pretty comparable to the healthy vaccinees and slightly better than recovered healthy donors who were SARS-CoV-2 infected. So basically, the conclusion from that is that at least in the short term, in other words, the first study that looked at natural infection and this study that looked at vaccination really only looked weeks to months after infection or vaccination. They could detect robust humoral and cellular immune responses in patients with inborn errors Caveat being, of course, if you don't have B cells, you're not going to make antibody response. The vaccines were safe, so vaccination should be encouraged for inborn errors of immunity in general. But obviously, these are early days, so we need to do much larger cohort studies and longitudinal studies to see what the durability and the efficacy of vaccination in inborn errors against SARS-CoV-2 really is. So I'll stop there, and I just want to acknowledge everyone that was involved in that international study that I was involved with. Again, particularly Isabel, Georgia, Kate Sullivan, Klaus Vornatz, and many others on this list, and the patients and their families. And I'll stop there, and I'll thank you again for your time, and I'll take questions if there's any. Thank you. Okay, good morning.
Hi, Kate.
Hola, buenas noches. My name is Kate Sullivan, and I am going to present in two parts. I have a little piece at the beginning that is 10 to 15 minutes long. Dr. Tanguy presented a little bit of the data, so we'll be able to go through that part a little bit more quickly. And then a colleague of mine is going to present a case that is really meant to be a cautionary tale. We don't have an answer for it, but I think the more people we vaccinate, the more we will see the rare and unusual consequences. As Dr. Bezrodnik talked about, there are adverse events with all vaccines. There's nothing unique about these vaccines. So let me start. I'm from Children's Hospital of Philadelphia. I'm a pediatrician, and my colleague, Dr. LaCava, is trained in internal medicine. And so why are we convening this? You can think about it really simply of the patients that are immune deficient. We worry if they're going to get protection. What will we do with our patients? How can we protect them in the face of this pandemic? And then on the other side of the coin, we want to understand, is there a group of people that should not get the vaccine because the risk of adverse events is very high? So that's really what I'm going to talk about with some data, some modeling, and some conceptual approaches. So this is a study that I really love. Actually, a friend of mine, Stanley Plotkin, put out a very similar study, but I like the graph in this one better. So you can find your favorite vaccine. I know every country has their own set or single vaccines that they're using. Let me explain how this graph is set up. So on the y-axis, this is the protective efficacy reported in clinical studies. Across the bottom, the x-axis is neutralization in vitro. So you take the serum from someone who has been vaccinated and you put it in a test tube with pseudotyped virus and you ask, does it neutralize? So that's sort of the gold standard rather than ELISA to look at antibody production. So what you see is an incredibly close correlation between the clinical efficacy on the y-axis and the in vitro neutralization. So at the upper right, you see the most effective vaccines, which are the mRNA vaccines and Novavax. And then way over on the left, you see the ones that are less effective. Sputnik did not appear in this study, so I can't tell you where it falls. So it says, in essence, that there is a strong correlation. You can almost predict the clinical efficacy based on the in vitro neutralization. Not that we're going to stop doing clinical trials. Of course, we will do clinical trials, but there is such a close correlation. There clearly, you can get a sense. And the reason I'm showing you this is the foundation for a set of models that were done by this group. So this is from the same paper, and it's asking the question, can you predict efficacy against the variants? Can you predict efficacy against the variants based on the in vitro neutralization? Sorry, my AirPods are running out of juice and I may have to switch over. So sorry about that ahead of time if I have to switch. So again, if you look at this graph, you're looking at efficacy against the wild type vaccine, and that's the dotted line. And then they imagine 3 different types of variants where you have altered binding in vitro. And based on the graph that I showed you before, they're predicting clinical efficacy. So why am I sharing this with you? Well, we all know variants are coming. They're already here, and there will be more. We're not done. It's not like Delta is going to be the last variant we see. And so we need to understand how effective the current vaccines will be against those variants. And so using the modeling that I've talked about, you can see at very high rates of efficacy with the vaccine, you don't really lose much. You don't lose much with the variants. But once you get to lower levels, the vaccines that are not as robust, they are not as strong, then you start to see decrements with the variants because you lose the in vitro neutralization, and that translates to compromised clinical efficacy. Now, the other question that Stu Tangi brought up that is certainly relevant for all of us is, what does this mean over time? So Stu presented data showing that immune deficient patients can produce antibody. The question is, will it last as long? So of course we can't predict in any one patient, but let's just look at the general population for a minute. So this is based on 3 different decay models. So there's only 3 sets of clinical data about the decay of antibody right now. That will change dramatically over the summer, our summer, your winter. But for right now, what we know is out to 6 months based on a study of natural infection of nurses in the United Kingdom, and then the 2 mRNA vaccine studies where they followed people out to 6 months. So based on those data, again, using this modeling system, you can see that out to 3, 4, even 5 years, you still have good efficacy against severe disease, which is the dotted lines, but you have much less efficacy against mild infection. And that is exactly my experience at my hospital. There have been a number of breakthrough infections, and they have all been mild. We all got vaccinated as part of a hospital program back in January and February, and there have been some breakthrough infections, but all of them have been mild. In fact, some were picked up through contact tracing. So that is my experience, and that is typical for other vaccines as well. As your immunity wanes, you retain protection against severe infection the longest, and you lose protection against mild infection. All right, so how would I summarize this? There's nothing really fancy other than I've shown you some lovely models that kind of make real the issues that we're facing. So more antibody is better, right? If your starting level is higher and you decay at a certain rate, you're going to end up with more in 5 years. So more antibody is better, but some antibody is better than no antibody. And then protection is going to be unpredictable. So everyone wants to know, do we need booster shots? What's going to happen to our patients? We don't exactly know, because we don't know what variant there will be next year. We're on Delta now. What are the future Gamma variants going to look like, and how will that impact the neutralization? So we can't predict the variants. And then all of us involved in immunology know that our patients can have unique trajectories. So there's the population average of the decay rate of the titers, but we know that there's a lot of variation, and our patients tend to lose immunity faster. So it may be that there are subsets of our patients that need boosters before the general population. And just as an example, in specific antibody deficiency, we know that some patients with specific antibody deficiency mount responses right after their vaccine, but they lose it in 6 months. And so that's just one example of how this personal trajectory might play out. Well, is there more than antibodies? So Stu Tangi talked about this a little bit. Let's just look at a tiny bit of data from mice. There are very similar data from other animal models, but I happen to like the way this study was done. So let me just explain it to you. So at the top, this is a mouse that is infected with SARS-CoV-2. The mouse recovers. In fact, mice generally recover from SARS-CoV-2. So this is a mouse that recovers from the infection. They take serum from the mouse and T cells from the mouse and put it into another mouse. that doesn't otherwise have any immunity. So now the only immunity in these second mice comes from what they got from the previously infected mice. And then they infect those mice and they ask what happens. So you're trying to dissect how much does antibody all by itself protect and how much does T cells protect all by themselves without antibody, which is of course impossible to do in people other than you could look at patients with XLA. So this is the control. So the black lines, the black bars here are the mice that got nothing. So the first graph that you see on the left, they're doing PCR for the virus, and the graph on the right, they're actually recovering virus and growing it. So if you look, the black bar is what happens when there's nothing transferred into the recipient mice. The red bars are what happens when they get T cells. So by PCR, it doesn't look very dramatic, but if you look at the virus recovery in the right-hand graph, remember this is a log scale on the y-axis, it's actually almost 1,000-fold less just transferring T cells. But of course, you get the best immunity by transplanting antibodies from the previously infected mouse. So what does this say? Well, antibody is certainly what it's all about in terms of protection from infection. There's no question that protection from infection is largely mediated by antibody, but for our patients that have XLA and no capacity to produce antibody, they will still make a T cell response to the vaccine, and that will protect them a little bit. Maybe not as much as antibody, but it will offer them some protection. So what does this mean for people with CVID? Well, they can make antibody, and some protection with even a little antibody production is helpful. Stu showed you the data about the patients with common variable that were vaccinated. I'll show you that as well. And certainly even lacking strong antibody production, they will still get T cell production. And so we are recommending that our immune deficient patients get vaccinated, even if they have an antibody problem. So this is the data that Stu shared with you. I do think it's really important. This is the study from David Hagen. And so I'll just remind you of the punchline. I won't go through it in the same level of detail. So the first column is what people look like before they got the vaccine. So the pink symbols are people before vaccination. So no antibodies, makes perfect sense. The dark red symbols are immune deficient patients post-vaccine. So Guillermo showed you what happens after 1 dose, after 2 doses. These are patients after 2 doses. And they compare it with healthy people after the same vaccine regimen and people who are convalescent from natural infection. So who responded? That's what we want to know. So Stu already pointed out there were a few that had no response. They were largely people with antibody problems. But look, most of the patients with common variable did respond. So what is my experience? Completely aligned with that. I have 3 patients with common variable who got the vaccine. They all responded. 2 patients with specific antibody who responded. And I have 1 patient with XLA who did not make antibody. So my personal experience exactly lines up with that paper. So what about risks? So none of the licensed vaccines or even vaccines that seem close to getting licensed are replicating virus. So they're not capable of causing infection. Yes, the coating is made from a virus in many cases, but all of them have been crippled. They do not replicate. They cannot cause a viral infection. So really what we're talking about is the classic adverse events that Dr. Bezrodnik talked about. We have to be mindful of that. Our patients will ask us. They are already asking about it. So what about interferonopathies? These vaccines induce really high levels of interferon, and so should we restrict our patients with a Cardi-Gouttiere? So we do. I know not every center does. And similarly, we do not give it to people with severe mitochondrial diseases because infections and fever can tip them into a metabolic crisis. But we certainly give it to the well-controlled older patients. So those are 2 subsets where we do restrict it. That's not very typical for our immunology clinics, but those are two unique subsets where we restrict it. But is there more? And so Dr. LaCava is going to present a case. I said a few minutes ago, we don't have an answer as to what happened to this young man, but we do believe it is vaccine related. And it points out what Liliana was saying, where we have to be very mindful about the adverse events, and we have to track them. So the rare events like the one you're going to hear about, those certainly aren't happening all the time, but we need to understand what is happening in those patients so that we can prevent it from happening again. So I'm going to stop sharing, and Dr. LaCava is going to present a case. Anthony, can you share your screen? Excellent.
Can everyone hear me and see me?
Yes, very nice.
Can everyone see me?
No, we just have an A.
I'm trying to start my video, but the host has stopped it.
Oh.
I'm not sure if they want to start my video if possible, but.
Liliana, can you help Anthony get his video started? And Anthony, maybe, there
Perfect.
You are.
Perfect, there we go. Good evening, hola, buenas noches. My name is Anthony LaCava, and I'm a fellow in allergy and immunology at the University of Pennsylvania. And I'd like to thank my colleague, Dr. Kate Sullivan, for inviting me to speak at this lovely talk. And I'd also like to thank Liliana and Ekaterini for arranging this wonderful conference. It's really special to take part of this cross-cultural transmission. And what I wanted to do was to take time to discuss a vaccine event, a patient that Dr. Sullivan and myself had taken care of at the Hospital of the University of Pennsylvania. And really, this is a case that we can all learn from, and is sort of a warning for all of you about possible rare adverse effects that can be seen with this vaccine. So I'll just start with the initial history of present illness. So this was a 20-year-old male who was previously well and received the first dose of the Pfizer mRNA COVID-19 vaccine. And within 2 hours of receiving the vaccine, he developed fatigue, vomiting, diarrhea, and lower abdominal pain. And then within 24 hours, he noticed whole body stiffness and urinary urgency. And at 2 days, he had developed gross blood in the urine, and he developed some tinnitus, some hearing loss, and had a pustular rash that erupted in his base and chest. And this was a patient that had never had a prior issue or any reaction to vaccines in the past. He had not had any recent sick contacts, preceding colds, any diarrheal symptoms, no new medications, no over-the-counter analgesic use or supplements, travel, recreational drug use, or any sexual activity. And actually, both of his parents had tolerated both doses of mRNA COVID vaccines. So upon initial presentation to the hospital, he was found to have a Tmax of 100.3 degrees Fahrenheit, and he was tachycardic in the 120s. He was with a normal blood pressure and with normal oxygenation on room air. And on physical examination, he was found to have an acneiform rash on his face, also on his chest. Though his lungs were clear, he was tachycardic. And though his abdominal exam was normal, he was found to have a pustular rash all over his face, his chest, his upper back. And though he had a normal neurological exam, he did have both ears, he had complete hearing loss. So some of the labs that had been obtained right on his initial presentation were pertinent for neutrophilic leukocytosis. He was lymphopenic. And as soon as he had walked in the door, his creatinine was 3.8 with a prior baseline of normal. So he was already in renal failure. He had a normal lactate, he had a normal INR and ferritin, but he did have some elevated inflammatory markers with an ESR of 29 and a CRP of 2.5. Workup also included HIV, syphilis screen, Epstein-Barr, CMV, hepatitis panel. Those were negative. And then he had a rheumatologic autoimmune screen, which was normal for ANA, ANCA. He had a normal LDH and haptoglobin. He had normal quantitative immunoglobulins and a normal SPEP. And the team did evaluated him extensively for infection, and the workup was negative. He had 3 COVID swabs that were normal. He was swabbed for flu, and that was negative. He had a rapid strep that was negative. His urine and blood cultures showed no growth. He had stool studies that were normal. He even had fungal blood cultures that were negative, and he had a lumbar puncture that was normal, and the CSF cultures showed no growth. Because of his hearing loss, he ended up getting an MRI of his brain, and that showed some punctate areas of ischemia in multiple vascular territories. And because of his kidney injury, he underwent a renal biopsy, which showed glomerular hemorrhage, which is usually immune mediated. And the skin was biopsied, and that showed suggestions of inflammatory folliculitis. His bilateral hearing loss was really of an unclear cause. So in summary, we had this 20-year-old male with no medical issues who acutely had a febrile inflammatory response after a vaccine, manifesting with rash, hearing loss, hematuria, and renal failure, with no pathogen that was identified and no signs of autoimmune disease. He did have an initial cytokine panel that showed an elevated IL-2 receptor, an elevated IL-10, and elevated IL-13, which is indicative of some T cell activation. And he also had an elevated CXCL9 level, which is indicative of gamma interferon effects. He also had a whole exome sequence that showed some variants of unknown significance in the C1s gene, though he did have normal complement studies. So his hospital course, so he had received high-dose steroids, plasmapheresis, IVIG. He had required renal replacement therapy. He also required pressors. He even developed a capillary leak syndrome with bilateral pleural effusions requiring a chest tube, pericardial effusion with tamponade requiring a pericardial window, profound anasarca. And then he also had developed alopecia areata. So what happened to this guy? So hearing loss is associated with therapeutic interferon, and alopecia is also associated with local interferon production and usually is treated with JAK inhibitors. Renal failure has also been reported with interferon treatment. Capillary leak syndrome has been reported with interferon therapy, and lymphopenia can also be caused by interferons. And we keep asking ourselves this question, like, why this guy? An article by Gogas et al. Published in Anti-Cancer Research in 2004 about adverse effects of interferon alpha treatment in oncologic patients had shown a profile of the type of side effects that these patients can develop. And really, our patient had almost all of these. So, we think that the mRNA vaccine had caused an interferon response, which ultimately contributed to his inflammatory state. So in conclusion, we don't really know why, but if we saw this again, we probably would initiate a JAK inhibitor treatment for this gentleman. Thank you.
Any questions for Anthony before he closes his presentation? No questions, Anthony. Would you please stop sharing your presentation?
Yep.
So we can. Thank you very much for all the great presentations. I'll start making the questions. I'll have to translate some of the Spanish written questions, so maybe it's not so good. The first question is to Professor Guilhermo. Armando Cruz questions, how much time after COVID? please. Well, how much time after COVID?
Hang on, I just got listener questions.
Okay, first for you, Tanji.
Oh, I've got a question. Yes.
I don't know if you have any more questions. Do you have? You had a question from Saul and you answered. Maybe you can answer alive for
Yes,
us.
sure. Can you hear me okay?
Yes.
Yes,
Great.
okay.
Great. So yes, Sol had a question about NLRP3 gain of function patients who you would think might be susceptible to hyperinflammatory consequences of infection, but they haven't seen severe COVID-19 in any of those cases yet. And there have been some big studies of large cohorts of PRD patients in Mexico recently and so on. So I don't have a clear answer to that, but I was just wondering if the patients are on JAK inhibitors, maybe that's enough to attenuate the severity of the disease, the intrinsic disease of the interferonopathy, but maybe that's also enough to sort of poise their immune system, particularly their innate immune system, to be ready to go and be protective. But I don't really know. But that would be my wild speculation.
Any more questions for Professor Stuart Tanguy? Do you want to say something else for us? Once we don't have any more questions for you?
Stuart or Kate, can the patients with immunodeficiencies with receive the vaccine, they can to suffer the new infection, you know?
I imagine there will be breakthrough infections, just as there will be in the healthy population. I'm not familiar with any of those cases yet. Kate's probably got a better visibility of this than I, just because there's many few people in Australia and many fewer cases in Australia. Kate?
Well, just because Australia's done better. So we have not had any breakthroughs of patients getting the vaccine and then getting an infection. So I'm sure it's happened, but none of my patients have reported it. So I don't have a good answer for you.
Okay,
may I read the questions? Here, okay, for Professor, first, Guillermo, how much time after the COVID-19 they measured antibodies, 3 months, 6 months, once the antibodies after COVID go in a certain time? And the other question is, how much time after COVID patients who had COVID-19 received the vaccine?
Bien, lo que hemos visto es que, bueno, además se ha publicado que los anticuerpos pueden durar hasta 6 u 8 meses, y si desaparecen no quiere decir que no haya inmunidad. Linfocitos B y T de memoria persisten aunque no hayan anticuerpos. Nosotros lo que hemos visto es que individuos que habían tenido COVID hasta 8 meses antes de empezar a vacunarse mostraron el efecto de intensificación con la primera dosis, lo cual habla que se está activando realmente linfocitos B y T de memoria. Y unos pocos pacientes que habían tenido COVID y no tenían anticuerpos, también vimos el mismo efecto con la primera dosis, lo cual nuevamente refuerza el concepto de que con la primera dosis lo que se está reactivando es la memoria, el linfocitos B de memoria. Y la otra pregunta, ¿cuál era?
When they gave COVID-19 vaccines in persons who had already COVID.
Sí, es eso. En los que ya tuvieron los que ya tuvieron COVID, hasta los 6 meses hemos visto nosotros, o sea, si tuvieron COVID 6 meses antes de la vacuna, el efecto de la primera dosis se mantiene. Reactiva la memoria de los que ya tuvieron COVID.
Okay. A next question is about the China vaccines. Once they are less effective, if people who received those vaccines are going to receive extra doses from another vaccines. What do you think about it?
Yo creo que la combinación de vacunas debería funcionar. Y inclusive dar una dosis adicional de la misma vacuna, como ha pasado con las vacunas chinas, creo que fue Sinopharm, en Estados Árabes, funcionó y reforzó la inmunidad. O sea, yo creo que sí, que debería reforzar, mientras el inmunógeno sea el mismo, se debería reforzar la memoria inmune, aunque sea distinto el adyuvante.
Anyone else wants to give an opinion about this?
We are using this vaccine in Brazil. It is frequent here. Actually, we are producing it under license of the Chinese government. And here, the efficacy is around 50%. So explaining again, it reduces in 50% the chance of getting COVID. However, it works very well to reduce the mortality. We have to remind that in Brazil, the campaign started on January with this vaccine targeting health professionals who work in the public service and the elderly. And for now, we are immunizing people around 40 to 50 years old. So the mortality rate dropped dramatically in the elderly who received this vaccine. So it really works, reducing the mortality.
Okay, there's another question that maybe all of you may answer about the recommendation for SARS-CoV-2 vaccines in patients that receive IVIG and sub-QIG. Anyone?
Catherine, are you asking about safety or efficacy?
I don't know, because there's it doesn't mention it. Just recommendation.
I think the securities, yes.
I don't know, because security, I think Kate spoke about it. Maybe it's the time for immunization between the application, the infusion of IG. I don't know, maybe.
So I'm going to take a stab at a different question. Since I'm not totally sure about that one, there actually is another model out there that I didn't present that shows that plasma derived from the US actually has quite high levels of antibody to SARS-CoV-2, both from natural infection and from vaccines. And the predicted level of antibody in immunoglobulin products is predicted to be therapeutic, meaning it will prevent infection this summer. And I know many countries in South and Central America get their plasma from the US or even the products from the US. So we are very close to having protection from immunoglobulin products. I know that wasn't exactly the question, but it's surprising to me how rapidly it has happened.
No, of course, and it's safe to do to vaccine patients receiving IVIG, and they should receive. You already mentioned that. There's Francisco Espinoza is saying that in Mexico, the fatality rate is higher because very few testing is done and only the severe cases are diagnosed. And the same as in Brazil and many other countries in South America, I think. Let me see if there's another question. No, I can go to questions and answer.
What is Kate, the one question is, what is your recommendation about the use of gamma globulin in the patients and the COVID? What are the moments with the patients receive the vaccines in the patient who receive the treatment with gamma globulin?
The timing.
Yeah.
So remember the data that suggests the immunoglobulin products will be protective, it's just a model. Until we have a lot of patients out there in a community where there's still active transmission, it's going to be very hard to know how effective it is. I have been recommending vaccination for my patients because we're not yet at the point where we think antibody in the products is protective. I don't think I will change my mind in the summer when we think the levels are high enough because I can't be sure. So I'm not sure what others will do. I know some places the vaccines are very precious, and so you might choose not to give them to someone if you think they're protected. But for me, at the moment, I'm still recommending the vaccine.
And would you recommend the vaccine between the doses of, I think maybe that's the question, between the doses 2 to 3 days before, 2 to 3 days after? I think this is the question.
Katerini, I have heard that a lot. So I do not. I give the vaccine when I possibly can, and I don't adjust it. And I don't think there's any reason to adjust it. I have not, but there are a ton. You might know that the Immune Deficiency Foundation is having their patient meeting and all the patients want to know the same thing. So I don't adjust. I think there are people who do, but I'm not sure how they come to the way of sequencing it.
Yes, the same.
There's a question about cellular response about 1 doses of immunization in persons who had already COVID-19 before getting the vaccine. If someone knows something about this. Maybe Stu had.
Can you repeat that? Sorry, Katerina, can you just say that question again, please?
Okay, if anyone knows something about cellular response about after 1 doses of the vaccine in persons who had already COVID-19 before this immunization.
So are you talking specifically about PID patients or just the general population?
No, I think it's general population. Oscar didn't mention PID patients.
I can't talk about cellular responses, but I have seen papers that have shown that vaccinating with a single dose of people who have been infected, naturally infected, really does boost the antibody levels. So I'm not that familiar with the cellular side of things, but the antibody response certainly does. So it's essentially, that's the old-fashioned way of vaccination. You know, the second exposure really boosts your primary exposure. So I presume, arguably, given that there are pretty good correlates between the T cell and the B cell responses, at least as shown in some nice studies that have come out, particularly from Shane Crotty's lab and others in various places, I would suggest that the same amplified response is seen for the cellular immunity in those cases.
Okay, thanks. And that's a question for Guillermo and for Liliana. The vaccines for SARS-CoV-2 with adenoviral platforms, do they generate immunity also to adenovirus infections?
It's a good question. I don't know, but maybe possible. I guess it is. It protects. I don't have the answer actually, but
I don't know.
It work.
And do you think there's a difference about the the length of protection generated by different platforms of vaccines?
Actually, we don't know so far, but I think that with the 2-dose vaccines, the protection may be longer than the single doses, but we don't know so far.
And what do the others think?
The easy answer, because it's in this moment it's difficult to answer this for me.
Condido, I guess.
I don't know. It's too early, I think, to be absolutely sure. And particularly, it's going to be the variants are going to complicate that data as well. So we just need the time to see what comes from those studies.
Okay.
Yes,
And
we need to study more.
And Agustina also asked about vaccines in Argentina. How are the situation about development of vaccines in Argentina?
Perhaps I mean
in this moment we begin to do 2 vaccines, Sputnik and AstraZeneca, and that is now in finally on June we are going to use our own production.
No. There's a question about
in June we are going to use our own production in this moment. Yeah.
There's a question
in the
about
future, different lab to do of the concept lab to do new vaccines. The
We can't hear. We can't hear. No te escuchamos bien, Lili.
Se corta, Lili.
Se me corta. Sorry, because I have the problem with the internet. In the in different lab, the concept lab, the research lab, we produce the own Argentina vaccines. Yeah, the San Martin University to do the new vaccines for our country.
And our group as well.
Yeah.
We are developing
AstraZeneca and Sputnik. Well, we share yet to do it. Sorry.
And there's a question about the thrombotic mechanism after AstraZeneca vaccine. If it's the same of thrombosis after heparin.
Yes, the patients present the antibodies en contra, el factor de heparina, ¿sí? 4 es el mismo mecanismo. Los pacientes, cuando hay es una clínica muy clara de trombocitopenia con trombosis, que aparece a los 30 días. Los pacientes no deben ser tratados con plaquetas porque se producen anticuerpos contra plaquetas y deben ser tratados con algunos casos con anticoagulantes. ¿Sí? Y obvio, no heparina ni tratar de usar con plaquetas, salvo que sea necesario.
Okay, Stuart, there's a question about NK cells response after COVID vaccines.
For me, I know.
Yeah, so that's really interesting. Obviously, there's been a lot of studies on adaptive immunity, T cells and B cells. There's been a lot of work on the innate system in terms of soluble readouts, cytokines, biomarkers, and so on. But the specific role of NK cells hasn't really been addressed, to my knowledge. Of course, I'm not pretending I'm an expert on anything, but I haven't seen any strong literature showing what the response of the NK cells is like at least during acute or convalescent responses. So that would be certainly an interesting area to study. We know there's the phenomena of memory in NK cells, more so for herpes viruses, not necessarily for coronaviruses. But it would be interesting to see what the functionality of NK cells is like in individuals who have had asymptomatic, mild, severe, hospitalization with COVID, and see if the NK cells have what they may or may not be doing in that context. But it's a bit of a undersubscribed area of research, I think, from the immunological perspective.
Thank you. And one final question for Kate. Do you measure autoantibodies against type 1 interferon in convalescent plasma for treatment? From Sara Espinosa.
So we have not been, largely because the turnaround time to get the results back is really long. So the assay is only done in one place in the United States, as far as I know, and they freeze the samples and run them. So it's very hard to use that information in real time because it can be a month before you get the results. So we have not been, but there's a lot of interest in, now that this is known to be a part of susceptibility to severe COVID, and because it strikes older men that otherwise don't have symptoms, there's a lot of interest in developing a commercial assay that would be more useful because you'd get results back quickly. So I think it may come because of the pandemic and because it could be used to direct your therapeutics in a more targeted way.
Well, we
Okay.
finished.
I think it's more than 2 hours, so I thank you very much. And Lily, please.
Well, thank you for your generous time. It's a very long webinar, sorry, but it's very interesting and you are very generous with us. Thank you. It's the honor to listen to you. Thank you. Gracias, Ricardo. Gracias, Antonio.
Bye.
Muchas gracias. Thank you. Bye-bye.
Bye.
Adios.
Pleasure.
Adios. Exactly.